everyone. In this video, we are going to discuss about the development of female gametophyte in angiosperms. In previous video, we have learned about the process of Megasporogenesis in angiosperms, and there we have seen that one of the cell present in the nucellus develops and grows into the megaspore mother cell. Okay, and then this diploid megaspore mother cell undergoes meiosis 1 and meiosis 2 and produces 4 haploid megaspores. Okay, it produces 4 haploid megaspore, and these 4 haploid megaspores, they are arranged in the form of linear tetrad. Okay, now for example, if this is the ovary wall, okay, this is the ovary wall, and there is a tissue called placenta. So we know that ovule is attached to ovary wall with the help of a placenta tissue. Okay, and from this placenta tissue arises the ovule. So first comes the stalk of the ovule, and after this comes the body of the ovule. Okay, now when we look at the body of the ovule, it's having these two integuments, okay, outer and inner integuments, and enclosed within within these integuments are the mass of cell which is called nucellus. Okay, so one of the cell of this nucellus towards micropyle, the end of the ovule, which is like, from where these integuments they appear to arise. Okay, so this end is known as chalazal end, and the end which is opposite to chalazal end is known as micropyle. Okay, micropyle end. Okay, now one of the cell of nucellus which is close to this micropyle end, it develops into the megaspore mother cell, and this one is diploid. Okay, and this megaspore mother cell undergoes meiosis process and produces 4 haploid megaspores. Okay, and these 4 haploid megaspores, they are arranged in the form of linear tetrad. Okay, linear means straight. While discussing about microsporogenesis, we have learned about the various types of tetrads like decussate, T-shaped, and then tetrahedral, isobilateral, linear tetrad, etc. Okay, so in angiosperms, always one type of tetrad is produced, and that is linear tetrad. Okay, this point is important. Note it. In angiosperms, only one type of tetrad is produced. Okay, whatever angiospermic plant is there, after the process of megasporogenesis, all 4 haploid megaspores will arrange themselves only in the form of linear tetrad. Okay, now out of these 4 haploid megaspores, the 3 megaspores, they will degenerate. Okay, so these last 3 megaspores, as you can see here in the picture, it will degenerate, and only one functional megaspore will be left, the one which is towards micropyle end. Okay, so the megaspore which is present towards micropyle, this will remain as such, okay, like a functional megaspore. Okay, and the other 3, the 3 which are towards chalazal end, they will degenerate. Okay, now what happens is this haploid megaspore, okay, this haploid megaspore which is left now is actually the first cell of female gametophyte. Okay, so this is the first cell of female gametophyte. Okay, now this megaspore which is the first cell of female gametophyte, initially it absorbs nutrients from the cells of nucellus and grows and develops into the size. Okay, so you can see here in this diagram, this megaspore now actually increases in the size. It actually undergoes growth, and nourishment is done by the cells of the nucellus. Okay, now this megaspore is haploid, and we know that all haploid cells, they undergo, if they have to actually divide, so they always undergo mitotic cell divisions. Okay, now one point to be noted here is, during the process of development of female gametophyte, remember clearly, first this haploid megaspore will undergo 3 successive karyokinesis. Okay, it will undergo 3 successive karyokinesis. That means successive means one after the other, without undergoing cytokinesis. Okay, without cytokinesis. Cytokinesis do take place, but at the end of the karyokinesis. So first this haploid megaspore will undergo karyokinesis by undergoing mitotic cell divisions. Okay, so this haploid nucleus is present after, like, three continuous mitotic divisions. This haploid nucleus present here finally forms the 8 nucleus. Okay, how many it will form? It will produce 8 haploid nucleus. Okay, so first nucleus after one nucleus after first meiosis, it will produce 2, 2 will produce 4, and 4 haploid nucleus. Again, after undergoing mitosis, it will produce 8 haploid nucleus. Okay, so in this female gametophyte, or in this haploid megaspore, I'm making a diagram here. In this haploid megaspore, after 3 successive karyokinesis, that means after 3 continuous mitotic divisions, what happens is 8 nuclei are produced. Okay, 4 of them are present at the each corner of the cell. Okay, now out of these 4 haploid nucleus at the each corner of this particular cell, 2 haploid nucleus move towards the center of the cell. Okay, so 2 haploid nucleus will move towards the center of the cell. Now it's like 3 haploid nucleus are left on each end, and 2 haploid nucleus are present in the center. So these 2 haploid nucleus which are present in the center are known as polar nuclei. Okay, and undergoes cytokinesis. Okay, the cell division. So what happens is this cell now undergoes cytokinesis, cell division, and it produces 3 cells at the chalazal end. Okay, because this end is present towards the chalazal or chalaza. Okay, chalazal end. So these 3 cells are produced at the chalazal end, and the end which is towards micropyle. Okay, so this end develops 2 synergid cells and 1 large egg cell. Okay, so the 3 common cells, the 3 similar cells present towards the chalazal end are known as antipodal cells. You can look here in this diagram. Okay, the nucleus present in the center of the cell are known as polar nuclei. Okay, and the 3 cells present at the micropyle end. Okay, so out of those 3, the 2 cells present on the sides are actually synergids, and one large cell present in the center is actually the egg, female gamete. Okay, that serves as an egg or the female gamete. So this is the structure of female gametophyte. Okay, now if you'll count this one, how many cells are present? 1, 2, 3 antipodal cells. Okay, then 1, 2 synergids. So 3 plus 2, 5, and then 1 egg cell, 6. So 6, and plus this one large cell. Okay, one large cell inside which all these cells are present. Okay, so there are total 7 cells. Okay, so how many cells are present in the female gametophyte? 7 cells. Okay, and let's count the nucleus now. There are 2 polar nuclei, so 2, 3 haploid nucleus of antipodal cells. So 2 plus 3 is 5, and then 3 more here. Okay, 2 nucleus of synergids and 1 nucleus of egg. Okay, female gamete. So there are total 8 nucleus. Okay, so female gametophyte in angiosperms is actually 8 nucleate and 7 celled. Okay, this is the important characteristic of female gametophyte. Remember this: the mature embryo sac or female gametophyte. Female gametophyte can also be called embryo sac. Okay, so the mature embryo sac of female gametophyte in angiosperms is actually 8 nucleate and 7 celled, and all these nucleus are haploid. Okay, so it is 8 nucleate and 7 celled. So I hope this entire process of development of female gametophyte or embryo sac in angiosperms is clear with you guys. If you have any doubt, please mention them in the comment section below. Okay, now let's quickly revise it once again from the next 2 slides. First, what happens is the diploid megaspore mother cell, which is present inside the nucellus, undergoes meiosis and produces a linear tetrad of 4 haploid microspores. Okay, and this process in which the diploid megaspore mother cell undergoes meiosis to produce 4 haploid megaspores. This process is known as megasporogenesis. Now, after megasporogenesis, the upper three megaspores which are present towards the chalazal end, they actually degenerate. Okay, they degenerate, and one megaspore which is present towards the micropyle remains functional, and this megaspore is actually the first cell of the female gametophyte or embryo sac. Okay, now this functional megaspore undergoes 3 successive mitotic divisions. Okay, free nuclear mitotic divisions, that means only karyokinesis. Okay, so after karyokinesis, or after 3 successive mitotic divisions, one nucleus produces total 8. Okay, so this is haploid nucleus. It will undergo first mitosis, it will give rise to 2. This will undergo second mitosis, it will give rise to 4, and this will undergo third mitosis, and it will give rise to total 8 cells in the 8 nucleus, 8 haploid nucleus inside the cell. Okay, inside this megaspore. Now there are total 8 nucleus produced, and 4 of these nucleus is present at each end of the cell. Okay, or each pole of the cell. Okay, now one nucleus from each pole moves towards the center. So this developing embryo sac is now having 2 haploid nucleus in the center and 3 haploid nucleus at each end. Okay, at each pole. Okay, now these 2 haploid nucleus in the center are known as polar nuclei. Okay, and the one which is present towards chalazal end. Okay, these now this actually, the cell after karyokinesis undergoes cytokinesis, and the separate cell membrane is developed outside around these haploid nucleus. Okay, so the cells towards the 3 similar cells towards chalazal end are known as antipodal cells, and the nuclei present in the center is called polar nuclei, and the 3 cells present towards the micropyle end. Okay, so 2 out of them, the one which are present on the sides, is known as synergids, and one large haploid cell present in the center is known as egg cell. Okay, that actually forms the female gametophyte egg cell. Okay, now 3 nuclei towards micropyle end constitute egg apparatus. Okay, so remember these 2 synergids. Okay, so remember 2 synergids plus 1 egg cell actually forms the egg apparatus. Okay, it forms the egg apparatus. Okay, and one more very important point here: synergids, they actually have thickening towards the micropyle end. For example, if this is a synergid cell, so towards the micropyle end, they actually have a thickening, and there are, like, small fiber-like structures, cilia. So this actually forms filiform apparatus. Okay, here it's written filiform apparatus. So filiform apparatus is important because it actually guides pollen tube towards the egg cell. Okay, it actually helps in movement of pollen tube towards the egg cell. Okay, and antipodal cells are the group of 3 similar cells present towards the chalazal end. Okay, now there are few points which are very important and you must remember. So remember, the mature female gametophyte or embryo sac of angiosperms, it is actually 7-celled and 8 nucleated. Okay, then now we know that this embryo sac of female gametophyte develops from a single haploid megaspore. Okay, hence this development of embryo sac from a single haploid megaspore is known as monosporic development. Okay, it is monosporic development. Mono means one and sporic means one single megaspore cell. Okay, and as this female gametophyte develops inside the ovule, okay, here the female gametophyte is developing inside the ovule, so it is known as endosporous. Okay, and remember, female gametophyte is colorless. It is endosporic because it is actually developing inside the ovule. Okay, and then it is this development is called monosporic development, and it is concealed inside the ovule within an ovary. Okay, so we have ovary. Inside ovary we have ovules like this. Okay, so inside each ovule we have a female gametophyte. Okay, so in next video we are going to discuss about the process of pollination.